So I started at EMBL at the end of 2009, so with the class of 2009. So the first impact with this institute was to gather and meet these youngsters, 25-year-oldish, junior class of PhD student, and we were approximately 50. And this was the first impact with an experience that changed my life in a way, because we started together a journey towards, learning to be a scientist. And I started in the lab of Christian Herring, and so my interest was focused on yeast genetics and also structural biology. At the time, Christian was in between the unit of structural and computational biology and cell biology. So in my PhD project, I was walking at the brink of these two fields, which I think it's a bit of a trademark of the EMBL, because in other places you will be more specialized in a sector of molecular biology, while here I could have a chance to discuss and to meet people with a very different background, which I think was quite unique.
Tell me a little bit about more what was your project about? What was the science? I mean, it's kind of peculiar to have structural biology with yeast genetics. So what was the science about?
Yeah, just if I may add one thing, I think that the two things were going together. So this community of students and the interdisciplinarity, because since we knew each other from the very first day, it was quite natural for us to discuss together about very different topics over a coffee. And the two things were working together. So we were building together a nice relationship, and at the same time we were discussing about our different projects. So this is one of my fondest memories, I have to say, of my PhD. And more specifically, since you asked about my project, so at the time I was very interested in studying the biochemical structure of a complex that is called condensin. And condensin is one of the molecular machines that organize the structure of mitotic chromosomes. And at the time, very little was known about how these subunits were working together. We knew that this protein bound ATP and was hydrolyzing ATP, and this had to do and manage somehow to condense the chromatin fiber, but how it worked, it was really a mystery at the molecular level. So the idea from the beginning was, we have this fantastic molecular biology and structural biology framework here, so why don't we try to do some structural biology working on this protein? But 10 years ago or a bit more, it was quite challenging to work with these huge molecular machines that are several hundred kilodaltons big, and cryo-EM was really still at its infancy. So we were trying to pioneer this together with other structural biology groups. But the focus of the group was not really structural biology, but more yeast genetics. So the idea was to combine this with mutational studies in yeast, and then figure out and link together the structural molecular aspect with the functional assay in this molecular model organism that is still a very valuable tool in the lab.
Cool. And I mean, what I like about it is that these are amazing molecular machines, and the approach of probing and mutating and changing so that you can then see what happens in the structure when you finally can see it. Certainly, it's a winning approach in understanding how the darn thing work, which is fascinating when you then come to the molecular understanding, because proteins are essentially entities that move and breathe somehow and do some molecular action at that scale. So that's very interesting. So then how did your career progress? I imagine you went for a postdoc. Did you stay on similar fields, or did you start exploring something different?
So the choice of my next step was very much influenced by the experiences that I had here, because I would say towards the end of the PhD, I got very interested in mass spectrometry, because it became quite apparent that cross-linking mass spectrometry was a very fantastic tool to be used in combination to cross these two kind of aspects, the functional and the structural molecular. and also for integrating structural information with data that would be useful to link and clarify what is the connection between the different subunits of protein complexes. And so I had the chance to get exposed a bit to how this technology worked and what were the principles, but I mean, I couldn't not go into the details too much. But then I decided to really go for proteomics for my postdoc and focus more on method development. Mm-hmm. And that's why I decided to move to Zurich, ETH, because at the time that was one of the best options to develop in that direction. And so I joined the lab of Paola Picotti in Zurich. And at the time, this was a junior lab. So I did not do the typical choice of many PhD students of going to the very famous big labs. Because I really liked what was the idea behind and what was the message that this lab wanted to develop and deliver. So in this lab, there was a strong interest in structural biology, but with a sort of new approach, because the idea was to try out very old school technique like limited proteolysis. So the idea was to try to make some nicks in protein structures, and then by using mass spectrometry, measure these fingerprints that could be associated to a certain structural state. And by doing this, we thought that we could use these signatures of different protein structure on a global scale to characterize different structural conditions of the proteome. So, for example, if you, let's say, have a situation where you have cells that are binding a certain small molecule, then the fingerprint of the proteome would be different. Or if you have, for instance, a disease where there is aggregation of a protein or a group of proteins, you will be able to measure that certain proteins undergo this kind of structural aberration. So this allows, by measuring a global fingerprint of the proteome, to read out different diseased states or functional states.
Oh, okay, so you were going from a single molecule focus to a very broad cellular state focus.
That's so the idea was a bit to try to do the same, with a lesser resolution, with a little bit lesser resolution, but have a global signature for all the molecular machines in the cells, potentially.
Yeah, and then proteomics is so fascinating because, I mean, there's so many flavors. I mean, starting from the classical stuff, going to these more recent applications, spatial proteomics, where you actually look into tissues, and all, of course, the modifications proteomics. So it's very, very, very exciting. And then of course, you became independent. So you established your lab. How was that, coming off, good experiences in PhD and postdoc? I was becoming independent. Did you find it a challenge? I did find it quite a challenge, but I wanted to hear about your thoughts.
Yeah, I think, for anybody who's starting this enterprise, I would say, expect something very different than being a postdoc or a PhD student. At least in my experience, I found the transition from becoming a postdoc, from being a PhD student, quite straightforward, I would say, because the type of work and your typical day is really the same. Or like, you are focused on your experiments, on your projects, and you're really fully into the science. Becoming a group leader is also a lot about organizing, managing people, and make them motivated. And there is also an administration aspect with it that everybody tries to minimize, but is definitely also part of the job. So I think the pleasure of doing this job is less of being directly involved in doing your own experiments, but seeing people developing and learning and managing to do things on their own, and appreciating also how they grow. So in a way, seeing yourself in them a few years back.
Yeah, no, I certainly agree with that. I mean, you start to develop a sense of things like building a legacy, you know, promoting careers, helping development of others, which I find personally very fulfilling and rewarding, as much as, you know, is rewarding great science and discovery. So, and now in your lab, what are you particularly focused on scientifically?
So we are trying to bridge somehow what I did in my postdoc and my PhD studies. So we still have a strong focus on technology development in the field of proteomics. So as you mentioned before, normally people think about proteomics as a systems biology science where you would catalog the different levels of proteins in a cells in different conditions. But instead we still try to have a sort of globular perspective on the molecular mechanism, so still focus very much on the structural aspect. So in a way, bridging structural biology and the systems biology aspect to it. So a sort of in-cell or global structural biology that is also something that other people are pushing a lot. But then we try to apply it to answer many interesting, I think, biological questions that have to do with the structure of chromatin and how gene regulation works. And so nuclear architecture and how factors like metabolite binding and the role of metabolism can influence gene expression by affecting the structure of chromatin.
Okay, so that sounds fairly complicated, because I mean you took the single molecular perspective that you had with the global view, and I can imagine that merging together and doing on top some technological development about it, it's quite challenging. But I think is the theme that one of the themes that fascinates me is that, of course, you need technological development to enable you know, science and science progress. And on the other end, you touched on something that I think is very interesting, that some of the technological development could lead to build a number of atlases of things, but and especially in this era of big data, essentially that might not get to a full understanding. So I mean, yes, you get a lot in terms of description of things, but then the hard part becomes to make some sense out of it, which I think is a problem, a general problem of a few of these system fields. So how do you feel about that? I mean, do you have an idea that with the proper technology we'll figure it out, or you think it's, you know, the understanding will always be a bottleneck? What's your... Because a lot of people think, you know, well, if we develop the right technology and we are able to do a good description, we will get understanding.
I think you have to balance the two things, right? Nowadays, it's somehow inevitable to try to develop new technology to explore angles that were dark before. Because, so for example, going into the microscopic scale, there are other fields apart from mass spectrometry and proteomics that made enormous progresses in the last 5-10 years and allow us to address questions that were impossible. So for sure, you need to have a foot in the pushing of the technology, but on the other hand, you have to remember, you know, what it's all about. So keeping a strong foot into biology. And I think one of the ways to do this is still being surrounded by a community of people that have a lot of interesting biological questions in different fields. That's why I also chose to join a research institute where there is a good balance between the two aspects. So I work in an institute where there are strong focus on molecular medicine. So I have colleagues that work in the field of oncology, neuroscience, cardiology, and many of them have specific questions related to, I don't know, angiogenesis or leukemia, or just to name a few, or I don't know, Alzheimer's disease, and they know precisely which proteins or which molecular mechanism is involved, and then we can partner with them and jump in and collaborate with them for addressing their specific issues and their specific
that is very peculiar, I have to say, because in a sense, I mean, what you described before is fundamentally basic biology, not particularly focused on that, but I think that having an interface on very focused, I wouldn't say translational, but application outputs with collaboration is always very important. I mean, it's this theme of trying to bridge the gaps between fields in meaningful ways, which becomes tricky sometimes. So the other question that I had was about your now you are independent and you had been in quite a few places around Europe, and you're originally from Italy. So can you tell us a little bit of how you feel about having moved into many countries? Is something that you would advise to young people? What's the positive side of it? What's the negative side of it?
I think it was a very positive thing, especially having done it when I was very young, because this allowed me to meet people from all over the world, get exposed to different cultures with different way of thinking, and discover things that you would not have imagined if you stayed in your home country or in your hometown. And last but not least, this also allow you to have a network of people that have similar interests than you, that are not only linked with you scientifically but also personally. So if you decide to travel a certain country for leisure, then it's like that you will have a friend that can show you around where to go.
No, that's true. I always have this feeling of being a little bit at home everywhere, and also the other feeling of being a little bit a foreigner everywhere, because of course every time you are in a different place, you sort of miss something from the previous place or from your home country.
Yeah, I think for scientific creativity you need this shuffling, right? At some point. Every few years you moved, and then you have a complete different combination and a group of colleagues that you interact with that they have a slightly different focus, or they think in a slightly different way than you, or let's say the philosophy of the place where you come from. So for example, you mentioned this before. I think that this passion for fundamental research will always stay with me, and this is one of the things that stuck on me from EMBL.
Okay.
So that's why in my lab we are still very focused on it, but of course we also try to expand a bit our horizons in more applied or translational research related kind of questions.
You received the John Kendrew Award, and which I guess is a very illustrious award. And how do you think how did you felt about that, and how do you think will impact you somehow now that you have been receiving this prize?
Yeah, I'm very proud to having received this award. Because I'm really fond of my experience at EMBL, and being recognized as a sort of memorable alumna, it's a great honor for me. Then I think the first impact would be being here today, and also getting a chance to get to know a bit more the activities of the Alumni Association at EMBL, because when you are at EMBL, you don't know much about what's behind the scenes and how important it is, this Alumni Association. Probably the only time I thought about it was at the very end, when I registered and I left my email address to be contacted in the future. And what was quite surprising last year when I was contacted by you is to learn how many activities and how big also this community is, and how many people like you, for example, are so much heavily involved and dedicate a lot of time to keep this alive and to keep people together. So keep doing this job. Maybe I will also get more involved in the future.
So thank you, Ilaria. This was a very interesting conversation, and it's great to hear about your paths and your history. I think it will be great for the alumni of the EMBL community. So really thank you for talking with us.
Yeah, thank you for inviting me. It is great to be here in Heidelberg again at EMBL, and seeing that certain things are always the same, but there is always a bit of change on the campus.